Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss an important technique of biotechnology called genetic engineering. Actually, DNA is a genetic material which contains hereditary information which is needed to create an organism. So the genetic informations are needed to create an organism, but DNA does not produce an organism, does not create an organism. So DNA are able to produce the proteins by the process transcription and translation. So the DNA get transcribed. DNA get transcribed. It undergone the process called transcription. So the DNA get transcribed into mRNA. That means messenger RNA. That means messenger RNA. So it contains some information. So that information is going to translated into a protein. So that mRNA undergone a process called translation. So that mRNA translated into a protein, and that proteins are able to produce an organism. That proteins are able to produce organism. So this process is called this central dogma of molecular biology. So this whole process is called this central dogma of molecular biology. So DNA are able to produce proteins, and that proteins are able to produce an organism. Then the specific DNA, that is the specific region of the DNA molecule which directs the synthesis of protein, is called this gene. Is called this gene. Once again, I repeat that statement. The specific region of the DNA molecule which directs the synthesis of protein is called this gene. Is called this gene. If we make the alteration in the nucleotide sequence of a DNA molecule which results in different form of proteins, it results in the change in the formation of proteins. It results in the change in the formation of protein. So this leads into either the formation of inactive protein or different protein. So this alteration make a changes in the protein synthesis. It may be a different protein or it may be an inactive protein. So genetic engineering or recombinant DNA technology or gene cloning is a collective term which includes several experimental protocols, results in the modification and transfer of DNA from one organism to other organism, and that is called genetic engineering or recombinant DNA technology or gene cloning. Then the recombination process is brought about by two processes. They are natural recombination process or conventional recombination process Then the second one is artificial recombination process. So in natural recombination process involves in closely related organism. The natural recombination process involves closely related organism. It does not involve unrelated organism. The natural recombination does not involve unrelated organism. Then it can be defined as the natural recombination is nothing but the crossing over process which takes place between two homologous chromosomes. So the exchanges of genetic material between the two homologous chromosomes is called as natural recombination process. Then the natural recombination process involves closely related organism. It does not involve unrelated organism. While in the case of artificial recombination process, it involves unrelated organism. Artificial recombination process involves unrelated organism. So, the genetic material of an organism is inserted into the specialized DNA molecule of another organism, give rise to recombinant DNA or rDNA. So, the genetic material of an organism inserted into a specialized DNA molecule of another organism gives rise to recombinant DNA or rDNA by using some specific agents, like vectors, or by using some specific instruments, specialized instruments like electroporation, then gene gun method, then liposome mediated gene transfer, liposomes mediated gene transfer, then chemical mediated gene transfer, and finally micro injection. So these are this is a specific agent and this is a specialized technique to transfer the DNA from one organism to another organism. Now I am going to tell about the steps which are involved in the transfer of DNA from one organism to another organism in order to produce the recombinant DNA. So please wait a minute. Let me explain the steps which are involving recombinant DNA technology. Here I have drawn a diagram which shows the steps which are involved in recombinant DNA technology. So first point, isolation of plasmid DNA and DNA containing gene of interest. So we have to isolate the plasmid DNA. So plasmid DNA are present in the bacterium. It contains the bacterial chromosome. In addition to that, it contains plasmid DNA. Plasmid DNA is otherwise called as extra chromosomal material, and plasmid has the ability to self-replicate. So the plasmid is a good vector. Which are used in the recombinant DNA technology effectively. So we have to isolate the plasmid from the bacterium. Then the gene of interest, we have to isolate DNA containing gene of interest. So this is the cell containing gene of interest. So we have to isolate the gene of interest. So this is the gene of interest. Then the gene inserted into plasmid. The second step, the gene inserted into the plasmid, and that step is called as insert. So the gene of interest is inserted into the plasmid. So this is the normal plasmid, and this shows the gene of interest inserted into the plasmid. So that the third point, plasmid put into the bacterial cell. So this gene of interest inserted into the plasmid, and that plasmids are put into the bacterial cell, Now that bacterium is said to be recombinant bacterium. Because it contains the recombinant DNA. So it contains the bacterial plasmid DNA, and it contains the gene of interest. Gene is nothing but a segment of DNA. So it contains the gene of interest. So the blue color shows the gene of interest. That is why the bacterium is said to be recombinant bacterium. Then the fourth step, cell clone with gene of interest. Now this bacterium contains this recombinant DNA. So along with the bacterial cell, this recombinant DNA can also multiply because it is a plasmid. I already told you plasmid can self-replicate. A plasmid can self-replicate inside the host cell. So along with the bacterial cell, the plasmid, that recombinant DNA, are also going to get multiplied. So we are able to find the multiple copies of recombinant DNA which are present in the bacterial cell, Then the fifth step, identification of desired clone. So we have to identify carefully the desired clone, which thing we are needed. So we have to identify the desired clone carefully. Then it has various applications. If we identify the desired clone, and they are used in various application process. So here the sixth point shows various application. Genes used to alter bacteria for cleaning up toxic waste. So here it shows, the diagram shows the oil spill in the sea, so that oil spills are digested by or removed by this bacteria, that recombinant bacterium, because it contains recombinant DNA. And here the seventh point shows gene for pest resistant inserted into the plant. So the gene of interest, the gene for pest resistant, get inserted into this plant, and this plant shows pest resistant, and this plant shows the pest resistant. Whenever the vectors are not involved in the desired gene, it can multiply by the PCR techniques. Whenever the vectors are not involved in desired gene multiplication, it can multiply by PCR technique, that is polymerase chain reaction. Then the multiple copies of gene of interest can inserted into the host cell protoplasm by injection. So we can inject the multiple copies of gene of interest into the host cell protoplasm by injection, and otherwise we can short into the host cell of protoplasm by shotgun method. So we can short the gene of interest into the host cell by shotgun method. So that's all about the steps which are involved in the recombinant DNA technology. So if you have any doubt, please clarify with me. Thank you very much for watching this video. Please prepare all the topics sincerely and show some dedication to your studies. So thank you very much for watching this video. Thank you.